Started.
Okay, we'll start. Hi. So I wanted to thank Dan for organizing this whole series, from which I've learned a lot, and for the opportunity to organize my thoughts around retinal pH. So the idea is that extracellular pH and the retina can provide some information about how the retina handles hydrogen ion from glycolytic metabolism. And we come at this from a physiological rather than a biochemical point of view. And so this is a somewhat different perspective than many people in the audience have. So I'm going to talk about how we measure hydrogen ion in the extracellular space, which is denoted by that little zero there, or O there. What the gradient of hydrogen ion looks like across the retina, a few comments about the effects of light, hypoxia, and metabolic acidosis. Then I'll talk about our model of hydrogen ion production and the production of hydrogen ion and its appearance in the extracellular space. That's Q sub H. And they'll just make a couple of comments about isolated retina measurements as well. So this complements the work we and others have done for a long time on oxygen. And if you're curious about that, we have a review in Progress in Retinal and Eye Research a few years ago, and there's also a talk from the International Ocular Circulation Society that you can find on YouTube. And so before I go any further, I need to thank the people who have mainly been involved in this. Andrei Dmitriev, who was a research associate working with me for about 10 years, and Lissa Pednick-Silver, who did the initial pH modeling, and a few other people. So the way we do this is that we work in the eye of cats, or since about 2006, rats. These are all anesthetized animals. And mostly we work in dark adaptation, but I'll show you some light adapted data as well. We tried, one reason for not doing mice is that the mouse eye is even smaller than the rat eye, and we can't really maintain normal physiological conditions in mice, whereas we can in rats and cats. So we try to maintain normal physiological parameters, and we take blood samples all the time to check that. And fundamentally, the technique is to use pH-sensitive microelectrodes. So one barrel of these is made by putting an ion-selective resin in the tip of the electrode, and that records hydrogen, a voltage proportional to the Nernst potential for hydrogen ion. But it also records voltages, just regular old voltages. And so you have to subtract out the voltage signal to get the pH signal. So the second barrel of the electrode is essential for subtracting out the voltage signal, but it's also really essential in our experiments for being able to record the intraretinal ERG so that we know where we are in the retina. So we put the electrodes through the wall of the eye and drive them into the retina. And the bottom picture shows a rat eye in proportion to a cat eye. So the rat experiments are considerably harder, but we figured out how to do it. So just to mention these ERGs, which are, as I say, essential for knowing where we are. The the ERG changes with depth, and this is not a new result. This is just what it looks like in a rat. So you have down here vitreal ERG before we penetrate the retina. But then as you go in, the ERG changes, and so the B wave gets bigger and then smaller, and the C wave continues to grow. But you know when you're through the RPE, because the ERG basically goes away in the choroid. And that's the amplitudes of the B and C wave are shown down here. So we record the ERG a lot. These are vitreal recordings from a cat now many years ago, where we were able to make the animals acidotic with hypercapnia. The B wave doesn't really change until you get down to about pH of 7.2 in terms of arterial pH. The C wave actually increases, and we understand why, but I'm not going to talk about that. And it changes probably earlier than B wave. But that's all I'm going to say about ERGs. The basic measurement is the hydrogen ion profile. So this is in the normal retina. We penetrate with the electrode through the retina in steps, recording the ERG along the way. And then we do a gradual withdrawal of that electrode, and that generates this profile of hydrogen ion as shown at the top. So I need to emphasize that this is extracellular hydrogen ion concentration. We're not inside cells here. The electrode tip is small, but it's probably too big to be inside cells. And so this is all plotted. All the pictures you'll see were plotted with more acidity up. But since people don't tend to think in nanomoles per liter, I've also indicated a couple of pH values on this graph. So in the outer retina, there is always a peak in hydrogen ion concentration. And the fact that there's a decrease both toward the choroid at 100% retinal depth and toward the vitreous at 0% retinal depth means that hydrogen ion is essentially produced there, and then it diffuses down these gradients toward the choroid and the retinal circulation in vitreous. Of course, at the back, it's transported through the RPE into the choroid, and we anchor these profiles by assuming that the pH we record with microelectrode is equal to arterial pH at the choroid, which is a pretty good assumption. In the inner retina, as you can see, the hydrogen ion concentration is higher than it is in the vitreous or in the circulation. And while it's clear that hydrogen ion must be manufactured in the outer retina, we can't really tell whether hydrogen ion is produced in the inner retina or whether it's just higher because it's diffusing from the outer retina. So I'll have more to say about that. So these are profiles through a cat and a rat. Those are both in vivo. Mice we can record from in vitro, and this is under conditions where it's perfused on the vitreal surface, and there's a very thin film of liquid in contact with air on the photoreceptor surface. So photoreceptors are up in this preparation. And in all cases, this peak in pH is is in the outer retina, just past the midpoint of the retina. So this is the outer nuclear layer. So the pH profiles are different than the oxygen profiles. These are several traces recorded in the same animal for hydrogen ion and in a different animal, but a particular animal for oxygen. In the oxygen profiles, the minimum tends to be in the inner segment layer. It's further back than the pH peak. And the other big difference about these traces is that in PO2, we see peaks in the inner retina where there are capillaries, or near where there are capillaries. So there are sources of oxygen and diffusion away from those. And you might have expected that we would see little troughs in the pH profile corresponding to capillaries also, but we hardly ever see those. And it's not because of the dynamics of the electrode, it's because they really aren't there. And I think what that means is that the capillary pH in the inner retina in animals is essentially takes on the value of the extracellular fluid. So the capillaries are probably a little more acidic than the veins or arteries, but we don't really know. So here's what light does to pH profiles. We're thinking of the peak as being production. So if it's glycolytic hydrogen ion production, you would expect glycolysis to be reduced in light-adapted retinas where the metabolism is lower. And in fact, we see that. So this is a rat set of profiles in the dark adapted retina in gray, and in the same retina where we've saturated the rods with light. And the same thing is true in cat. So particularly in cat, you can see that the inner retina doesn't look much different in light adaptation and dark adaptation. And probably that's really true in the rat as well. So we think, and there's evidence of this from oxygen recordings, that in terms of steady light, the metabolism is the same as it is in darkness, which is certainly not true of photoreceptors.
I'm sorry, I thought that you had done a couple years ago an ex vivo preparation where there wasn't sort of an influence of circulation, and in that case, oxygen was affected by light and consumption maybe increased in the inner retina with light. Is that a misinterpretation on my part?
Yeah, so the distinction is that oxygen consumption doesn't increase with steady light, but it does increase with flickering light.
Okay, I thought one of the takeaways from that paper for me was that maybe there was a slight effect of steady illumination as well, but I'll need to reread.
No, there's a very small effect of steady illumination, which we were able to measure there, but we've never really been able to measure in vivo. Yeah, so it's a small effect of steady light, which is an increase in metabolism in the inner retina. But if you flicker light, the increase is much larger. We have not tried flickering light really with pH electrodes. The signals are noisier and smaller, so we don't really know about that. But what I want to emphasize here is that these changes going from dark to light are pretty slow. Um, Over here on the left is what happens in a rat as you expose the rat to 3 minutes of light at rod saturation. It takes 3 minutes to get down to the pH of the light-adapted retina. The inner retina is a little more complicated, and we discussed this in the review paper about this, but I'm not going to go there today. The same thing is true in cat, where it takes, you know, 2.5 or 3 minutes to change metabolism in the cat retina. These two traces are from different papers by Yamamoto. He was the first person in Steinberg's lab to really make these pH measurements in the intact retina. This picture at the bottom is a log unit brighter than the one at the top. And the main effect of that is that the recovery is much slower because you have a rod after effect. The rods are still hyperpolarized after the light goes off. And so the metabolism is still lower until it gets back to the dark adapted state.
How does the time course for changes in, oh, maybe you'll respond to that right now.
Yeah,
Actually, I was just going to compare. Yeah, go for
right.
it.
Yeah, it's a good question. We're used to things speeding up with more light in terms of electrophysiological activity. So the A wave, for instance, speeds up a lot when you make the light brighter. Ganglion cell responses all have a shorter latency when the light is brighter. These metabolic responses don't really change with with uh, with the brightness of the light. What they do is get bigger. So here again, this is a set of traces from Yamamoto et al. And with the brightest light, the time course is really pretty much the same as it is with 3 or 4 log units less light. And the interesting thing, as Dan is going to, is that this is really quite different from oxygen. And we believe that these oxygen traces, where PO2 is going up in the inner segment layer, we really believe that these really reflect the time course of the change in metabolism. And the time course is a little different in different animals, but in cat, the time constant of these responses is about 10 seconds. And again, there's this rod after effect, which with very bright lights. So the decrease in oxidative metabolism that you see with PO2 electrodes seems to be much faster than the decrease in glycolytic metabolism that you see with pH electrodes. So I don't know if that resonates with anybody or whether that's a surprise, but that's what we find.
Hey, Rob, I have a question. So how much, I mean, this must be influenced by the buffering capacity of the extracellular fluid, right? Is it mostly bicarbonate buffering, and is that something that can be manipulated like in your ex vivo experiments?
Yeah, so I'll show you how we've manipulated that in a few slides. Extracellular buffering is, I think, almost entirely bicarbonate. There's not that much protein doing buffering in extracellular fluid in the retina or really any place else. We never really looked at the time course when we blocked carbonic anhydrase. So that would be something that somebody could do in the future, but not us. So before I show you that, let me just take you through what hypoxia does. Hypoxia is well known to increase glycolytic metabolism in the retina, and it changes pH as well in the expected direction. So these are CAT profiles under normal conditions and then during fairly severe hypoxia. And the pH in the outer retina goes down to around 7. You can think of this, so these are now moles per liter, but this is 80 nanomolar, 90 nanomolar, so on. And the time course of this is actually reasonably fast. It's faster than the light evoked changes. So this is, again, from Yamamoto, one of Yamamoto's papers. In the dark, deep in the retina, in the outer nuclear layer, the hydrogen ion goes up with hypoxia. Light suppresses that, presumably because the metabolism's already lower, so it doesn't need to get pumped up. It doesn't need to get increased during hypoxia. It doesn't need to decrease further during hypoxia. Sorry. Need another cup of coffee, I think. And then you go back to the dark and the hypoxia response is restored. And they're a little bit bigger if the hypoxia is lower. And you begin to see an effect of light. We've also looked at metabolic acidosis. We never really looked at hypercapnia, but we looked at metabolic acidosis. And what this is plotting is the choroidal PO2 and the maximum pH in the retina, maximum hydrogen ion concentration. So it's the peak of these curves against arterial pH. So when you make the animal have metabolic acidosis, of course, the arterial pH acidifies by about 24 nanomolar on the average. But the retinal hydrogen ion concentration goes up more than that. So something is going on in the retina in addition to what's going on in blood. It's not just elevated by the same amount. What did I do there? Okay, so we tried to understand what the production of hydrogen ion was in the outer retina. So this is very similar to the oxygen model that we've used for many years. And this is Lissa Padnick-Silver's work. This model is a little more complicated because we're accounting for hydrogen ion going only in the extracellular fluid, whereas we can consider the retina continuum for oxygen. So here we have to take into account the extracellular volume fraction and the tortuosity of the extracellular space. So this follows the work of Charles Nicholson, who did a lot of these experiments in the brain. So the basic equation is this one-dimensional steady state equation for hydrogen ion diffusion and production. And the solution of that for each layer is the concentration of hydrogen ion is related to quadratic in distance plus a linear term plus a constant. And that Z factor is what includes the hydrogen ion production. And so the full model has 3 layers. So there's equations for the outer segment layer, the inner segment layer, and for the outer nuclear layer. We determined that there were 3 layers. And the interpretation of the curves is that if the curve is a straight line through one of these layers, then you've only got the AX plus B term, and there is no consumption in that layer, or no production in that layer. If the curve is a little bit convex or very convex, then that means that there's hydrogen ion production in that layer. So there's no production in layer 1, but the profile has a quadratic shape in layers 2 and 3, implying that there is production there. And so what we did was to fit the model to actual profiles of hydrogen ion. We've only really done this in the cat retina. And I'll show you some values later. So that's the model we have for the appearance of hydrogen ion production in the extracellular fluid in the outer retina, in the subretinal space, basically. So again, I have to emphasize that the model applies only to the extracellular space, not the intracellular space. And so the net rate of hydrogen ion production must be a balance between the gains of hydrogen ion and the losses of hydrogen ion. And so when we think about what those gains and losses might be, we can consider several possibilities. First of all, there's the sodium-hydrogen exchanger, which would transport hydrogen ion into the extracellular space. There's the MCTs, and they will transport lactate or other monocarboxylates along with hydrogen ion into the extracellular space. And there's a possibility that hydrogen ion is produced in the extracellular space by buffering of CO2. The only extracellular carbonic anhydrase in the retina is carbonic anhydrase 14. But of course, like all carbonic anhydrates, it facilitates the reaction in both directions. The possible losses of hydrogen ion would be uptake of hydrogen ion with lactate on the MCTs into other places, for instance, into the end feet of, into the distal end feet of the Mueller cells. And that would reduce the hydrogen ion concentration. And then it's also possible that hydrogen ion is lost because it's turned into CO2. Probably both of these buffering things are not going on. So the question is, which is it? But because the profiles are convex, we know that the gains must be greater than the losses. And there could be other hydrogen ion regulating mechanisms. For instance, electrogenic sodium bicarbonate transport into Mueller cells, which Newman has shown exists. There also could be chloride bicarbonate exchanges as well. But these complexities mean that we don't really expect hydrogen ion production to equal lactate production in the extracellular space. And I'll come back to that point. It might be kind of proportional to lactate production, but I'm not sure we're guaranteed of that. So the model is only about the extracellular space, and it doesn't reflect the losses of hydrogen ion through the boundaries of that extracellular space. So across the RPE or into the inner retina. And there is loss of the hydrogen ion in both of those directions, as the gradient shows. And at steady state, that production, that net production has to be equal to the total flux through those two boundaries. And we can calculate the flux through the boundaries because it's just the diffusion coefficient of hydrogen ion times the gradient. And because the gradient is steeper in the outer retina, we know that more of the hydrogen ion has to be lost through the RPE than is lost into the inner retina. But we haven't quantified either of those. So here's the model that we put into the review paper of pH that we wrote recently. And I'm not going to emphasize this because we did not, in that paper, try to come to grips with how this model fit with the actual mathematical modeling that I just showed you. And so that's what I'm trying to do here, although we can't really do it very quantitatively. So what I've done here is I've just emphasized that extracellular space, subretinal space, where we're accounting for the production of hydrogen ion. And I felt I needed to add a Mueller cell because part of the Mueller cell is in the outer retina, and I needed to allow for hydrogen ion and lactate to be uptaken by the Mueller cells in the outer retina. So again, the gains would be transport to the extracellular fluid by the MCTs from the photoreceptors. And the losses would be uptake by the Mueller cells, again on MCTs. And I've emphasized here that one loss is likely to be the buffering of hydrogen ion into CO2. And I've left out hydrogen ion transport to the extracellular fluid by the sodium hydrogen exchanger, and I've left out the opposite reaction of CO2 going to hydrogen ion, because there's some evidence that those don't happen in the outer retina. And now I'm going to show you the evidence that we have that these are negligible as far as hydrogen ion is concerned. So if the sodium hydrogen exchanger pumps hydrogen ion into the extracellular space, and sorry, this will be energy in the sodium gradient. And if you block that exchanger, then you would expect the extracellular space to alkalinize because less hydrogen ion is being pumped into the extracellular space. But using the rather specific blocker cariparide, which we gave intravitreally in rats, you dramatically increase the hydrogen ion concentration in the extracellular space. So the hydrogen ion is evidently not, the net effect of the sodium hydrogen exchanger is not to alkalinize the extracellular space. Probably what's going on is that the sodium hydrogen exchanger is important in moving hydrogen ion into the circulation, but not causing acidification. So in the paper we did on metabolic acidosis, we actually were able to localize the sodium hydrogen exchanger, and it's not out here where the photoreceptors are, although we can't really see the subretinal space here very well. But it is associated with blood vessels in the both the deep capillaries and the superficial capillaries of the retinal circulation, and it's closely associated with the vessels stained by this lectin.
And if we were to have seen the RPE here, we would also see some of that exchanger. Is that right?
So the slides we have, and I should go back to Dave Antonetti and see if he has slides that show that. It should show up in the RPE, but I don't know if it does or not. Now, there is some evidence that photoreceptors have the sodium hydrogen exchanger. That's from a paper by Katz and Oakley. So it may be there, but it's not as important as the transport into the circulation, at least as far as extracellular hydrogen is concerned. Here's what happens if you use a carbonic anhydrase inhibitor. So the first evidence of this was, again, from a Yamamoto paper. If they had the electrode in the inner retina and gave intravenous acetazolamide, they saw really nothing happening. But if they had the electrode in the subretinal space and gave acetazolamide, the extracellular space strongly acidifies. And we did that experiment in rats, and here's the profiles. So here's these green profiles are in the retina before the dorzolamide. After dorzolamide, there's a strong acidification of the extracellular fluid in the outer retina. And significantly, so when you give these carbonic anhydrase inhibitors, you could be interfering with both intracellular carbonic anhydrase, which is CA2 in the retina, as I remember, or CA14. But benzolamide was developed in order to only block extracellular carbonic anhydrases. It does not cross membranes very well. So when we gave intravitreal benzolamide in cat, we also got this very strong acidification down to pHs, around 6.5. So this suggests that the directionality is for hydrogen ion to be turned into CO2. And when you block that, you just wind up with more hydrogen ion. So that's why this picture is the way it is.
Well, so Rob, another question. So along lines I was asking before. So if you, in the presence of the carbonic anhydrase inhibitors, does that change the kinetics of the change in pH in response to light?
Yes, I wish we had done that experiment, but we didn't ever do that experiment.
The prediction is it would change faster, right? Because you lose your buffering capacity. Is
yeah.
If it's going from hydrogen to CO2, then the buffering, then it should.
And the CO2 would be lower because oxidative metabolism is making less of it.
Yeah. So you're changing the amount of buffer and the ability of the buffer to buffer.
Yeah. So it's complicated, and I wish we had done that experiment, but it's too late now. The other thing to notice is that if hydrogen ion is converted to CO2 in the outer retina. And of course, CO2 is faster to eliminate, easier to eliminate. That would require a source of bicarbonate in order to do this buffering. So it could be, and we don't know, that the buffering works in the other direction, in the inner retina. So if the inner retina is doing mainly oxidative metabolism and producing a lot of CO2 and not very much hydrogen ion, that CO2 could be converted into hydrogen ion in the inner retina, and that bicarbonate could then be used to help buffer the pH in the outer retina. But this is pretty speculative, and there's just no evidence. But blocking carbonic anhydrase really doesn't either acidify or alkalinize the inner retina very much. So this is evidently less important in the inner retina. The other thing that I worry about a lot is whether we're totally missing other sources of hydrogen ion here in the outer retina. And I don't think buffering by protein is likely to be very important. Buffering by other things that you have in blood and extracellular fluid is hemoglobin is of course not important. Anyway, that's where we are. So what do we learn from this modeling? Well, we've learned that there are two regions that essentially produce hydrogen ion in the outer retina, the inner segments in the outer nuclear layer. We've learned that light basically halves the production of hydrogen ion in the outer retina. This is data from one of Lissa's papers. That if you go down to breathing 10% oxygen in the cat retina, that will increase hydrogen ion production by about 2.5 times on the average. And the absolute values that we get are pretty interesting. So we think we've done the modeling correctly. We checked this many, many times. And the hydrogen ion production in the dark is only about 3 nanomoles per liter per second. So this is the net effect of all those gains and losses. If you measure lactate production, and Wang et al. Did this in the cat in vivo by doing AV differences in the choroidal circulation. Very difficult experiment, a lot of variation in the numbers, but they came up with about 49 micromoles per liter per second. So orders of magnitude higher than what we see with hydrogen ion production. And the hydrogen ion production is presumably coming mostly along with lactate production. But there are all these complications with hydrogen ion. If you try to look at lactate production in the isolated retina, I had trouble finding numbers for this, but from Winkler's paper in 1981 in rat, the number was really pretty similar to what Wang found in vivo. So the lactate production and hydrogen ion production are several orders of magnitude different. And I'm saying this is an underestimate because part of the lactate is not ever getting out into the blood supply. It's being used in the inner retina. So you're underestimating the amount of lactate that the photoreceptors are actually producing. So the absolute values of these are very different. And the question is whether they scale proportionately. So if you see an increase in hydrogen ion production by a factor of 2, does that mean that lactate production went up by a factor of 2, or are they dissociated? And this is interesting because the ECAR values that you get from seahorse type experiments are extracellular acidification. So they're very comparable to what we get with our hydrogen ion production values. They should be kind of comparable to that, although the units there are so funny that I can't do the conversion, or I couldn't do the version yesterday when I was trying to work this out. Anyway, those ECAR values are not lactate production, and so we have to take them with at least 1 grain of salt, I think. There are other reasons why we might do that too.
And Robert, another question. So when if the lactate goes out of the cytoplasm of the photoreceptor, it takes a proton with it and it releases it. Has a very low pK, so it releases it when it gets into the extracellular space. But then if that lactate is used by another cell, like a neuron in the inner retina, it's going to take that proton with it, right? So
Right.
so that proton would never, even though there was a lot of lactate released, but then it was consumed, that proton doesn't really get counted in terms of acidification of the extracellular fluid.
Right. I think that's right. Yeah.
And a kind of related statement is just, it's not just lactate that MCTs are handling, right? It's other metabolites that they're transporting. Arguably, some of those metabolites are far less abundant than lactate, so I do understand the focus on lactate. But I mean, Nancy knows this, and I was expecting her to sort of talk about it earlier, but they handle a wide variety of substrates.
Yeah. So this more complete mechanistic understanding of where hydrogen ion comes and goes would need to include those other monocarboxylates. And I personally don't know what the relative amounts of all of those would be. Yeah. So, I mean, I think it's an interesting subject for somebody to tackle. Maybe if somebody wants to give me some numbers, I'll tackle it some more. But we can't do any more experiments because the lab doesn't exist. So we're done. I just want to make a couple more comments here, and then if you want to know about pH during hyperglycemia and diabetes, we'll need another session for that. So I just wanted to point out that in any in vitro system, there are substantial unstirred layers. So these are experiments that I thought I had these are experiments that Andrei Dmitriev did when he was in Stuart Mangel's lab. And these are with the retina mounted in a sort of traditional chamber with perfusion on both sides. As he approached the retina from the bath, the pH went down. So the retina is considerably more acidic than the bath in cold-blooded retinas as well. And then as he backed the electrode back out, the pH came back up to the bath value. So, but there's hundreds of microns of unstirred layer here. This would be true for oxygen or any other metabolite or substrate that you're putting into the retina. There's a big unstirred layer which will influence the diffusion into or out of the retina. Same thing is true in the rabbit retina. The other thing that Andrei learned from the isolated retina was what happens if you do some metabolic poisons. So not too surprising, but in iodoacetate, which blocks glycolysis, the retina, which initially was pretty acidic, alkalinizes, not quite up to Ringer pH, but he didn't leave it. This is 25 minutes. So that's not too surprising. If you block oxidative metabolism with cyanide, and presumably turn up glycolytic metabolism, that can acidify the retina. These, believe, were in fish retina. If you use Wabane, it has a much smaller effect, because evidently a lot of energy metabolism still has to go on when you're in Wabane. And if you put it in glucose-free Ringer, the effect is very slow, presumably because all these other substrates come into play. Anyway, it's an interesting result that probably you might not know about. So that's what I have to tell you. Happy to have other questions or comments or.
All right, the floor is open. I guess a pretty basic question is whether or not you've kind of looked into literature to see the extent to which some of these extracellular pHs are actually affecting enzyme function or protein function in general. Is there kind of a consequence to maybe if you have metabolic acidosis and the fact that the retina is even still further acidifying things, how that might impact the whole system?
Yeah, so we don't know very specifically. What we referred to in the review paper that's in Progress in Retinal Research a couple of years ago, we referred to a lot of the things that can happen that are related to pH, including enzyme function. What we tried to do was to see whether acidosis would change the function of some of the acid regulating mechanisms in the retina, to see whether the retina could compensate for acidosis. And that was a little disappointing. We found changes in some RNAs, particularly the sodium hydrogen exchanger. We saw little changes in carbonic anhydrase and ASIC, but we did not really see changes in protein for any of those things. And this was after 2 weeks of metabolic acidosis with ammonium chloride in the drinking water. And so we think that if they had changed, they would have changed in that time. So we don't think that the retina is really capable of handling acidosis much better after a long period of time. But there are certainly lots of things that could be changing transiently as you go to a more or less acidic state. We just don't know anything specifically about which things would be most sensitive to that.
Jason has a question, and then I have at least one more.
Hi, Rob. Thanks for the talk. As you probably know, there's a phenomenon, especially in retinitis pigmentosa, where you get cysts of fluid, mainly in the inner retina, that seem to respond exceptionally well to carbonic anhydrase inhibitors. It's one of the we obviously are supposed to use those for glaucoma, but this is our off-label use for it. I was like just scratching my head trying to think of mechanistically how that happens. There's been lots of theories out there. I at first was hoping to hear that the protons would be more affected in the inner retina, or at least equally in the inner retina, but they're not, and that's where the fluid tends to be, although it's in the deep inner retina, sort of, in the INL. Anyhow, do you have any hypotheses on mechanistically what might be the links between this carbonic anhydrase inhibition and the drying up of intraretinal fluid?
I did think through that once, but I actually can't remember right now. So I was very puzzled by it seems to me that from our work, when you block carbonic anhydrase, it's always bad, and it just acidifies the retina. How acidifying the retina could remove fluid. Well, so there's also data from, I think, Sheldon Miller's lab that acidosis can influence the rate of pumping across the RPE.
So maybe that's the answer. Is just I'd have to look at the specific ion channels and how protons would affect that. But
Yeah.
You're saying it could be a drying effect all the way from the RPE sort of across the retina from the excess production of protons sitting there.
Yeah. So of course, that's the way the retina is dried out normally, is by pumping a fluid across the RPE into the choroid. And so if you can enhance that in any way, things are going to help. So it may be producing retinal acidosis, but there's a net benefit to that.
Thanks.
Yeah, and there are effects of the carbonic anhydrase inhibitors on glaucoma, as you mentioned, and those are presumably good. So dorzolamide that we used is one of those things that's used to bring down intraocular pressure.
Yeah, I mean that mechanism is well worked out. That's the water follows the proton sort of flux in the ciliary body, and so anyhow, so maybe it's presumably a similar mechanism in the RPE, since obviously both are epithelial cell types that may share a fair number of channels. Anyhow.
Yeah, I'll have to work that into my model. Thanks for reminding me about those experiments.
Okay, I have at least I guess when I was looking at your figure for the sort of peak area of acidity, I guess, or proton production, it seemed in my mind at least slightly offset from the peak area of oxygen consumption. Is that correct? And does
Yeah.
that imply that
No,
the synaptic metabolism in photoreceptors is substantially more glycolytic than inner segment metabolism? Or is there what are the takeaways for the sort of different parts of the photoreceptor in your mind?
Yeah, so we know from like work in the 1950s by Lowry and colleagues that the glycolytic enzymes are further proximal than the oxidative enzymes. And I should also point out that it's not necessarily that the acid is produced intracellularly in the cell bodies in the outer nuclear layer, but that's the point at which it exits into the extracellular space.
Sure.
So it's not necessarily the same as where it's all produced.
Yeah,
So
all
I
the
mean,
glycolytic enzymes, all like the hexokinase 2, PKM2, they all seem to be in the inner segment from imaging data that that plenty of labs have produced. That's kind of where, but your point is well taken.
and where is the MCT along the cell?
Nancy, are you still here? I don't remember. That's a great question. I mean,
Yeah.
that seems crucial.
Sorry, what's the I'm here. Sorry.
The question is,
I was looking up things, Rob, old papers, because I was jarring my mind. But anyway, Dan, what was your question?
Where on the photoreceptor are the MCTs localized?
Inner segment. I mean, inner segment and down the whole cell, including the synapse. And I'm just forgetting the disease. There is a William Sly paper that I was trying to find, and something we did talking about pH and the effect on both phototransduction. So when he has a mouse model where they knocked out carbonic anhydrase 14, it's a PNAS paper, and I was just trying to find it when you asked me if I was still here. I was trying to multitask to add something. Yeah. And also, I mean, Sheldon and I talked a lot about whether, because there was this Zoythan, years ago, probably early 2000s, published a paper where he showed that lactate could drive fluid movement. So the idea that we had that was, between one and on the apical MCT1 on apical membrane and MCT3 on the basolateral, that it was not only transporting lactate, but also water and protons. And so that the concentration of either protons or lactate could drive the fluid flux was the theory.
Yeah, but I guess
so I'll send you those papers, Rob.
Great. I think I may have them, but
okay.
But thank you very much. Yeah, I should say in response to Dan's earlier question that there's a lot of evidence that acid influences photoreceptor function by the groups in Finland mainly. And that's not recent work mostly, but we refer to some of that work in our review paper.
And Ed Pugh also looked at rate of phototransduction and extracellular pH, right, Jim?
Yeah, that's right.
Okay. Sorry, I just have to go back decades in my brain to pull out these things.
But that was the original phosphodiesterase assay with this production of protons. Paul Liebman. Yeah.
Oh, right. Okay. Thank you.
A local for you. Yeah.
Yeah.
Anybody else have any questions? All right, let's thank Rob again. This was such a great talk. I really enjoyed it.
Thank you very much.
Thank you. Thanks.
All right, and everyone have a great day.